Today we have Professor Taekjip Ha here to talk about experimental biophysics. He's one of the real pioneers in applying physics concepts to biological systems. And so he's addressed a lot of really interesting questions and made a lot of fundamental observations in the field of biophysics, molecular biophysics. And he came here in the year 2000. He had his PhD in 1996 from University of California at Berkeley. And one of the things that I really enjoy about this program is, for those of you who come frequently, you know this. Every 2 weeks I stand up here and I say, we are so fortunate to have someone here who is outstanding, one of the top researchers in the world in such and such a field. And it really is, it just reminds me how spectacular this department is. This is really one of the top departments in the world, not just the country. And it's no accident that week after week we have speakers who are really outstanding, very top researchers in the world. And let me just give you a brief rundown of a few of the awards that Taekjip has won in the last few years. Maybe this will give you an idea of what I'm talking about. 2001 Searle Scholar, 2001 Research Innovation Award, 2001 Outstanding Young Researcher Award, 2002 NSF Career Award, 2002 Fluorescence Young Investigator Award, 2003 Sloan Fellowship, 2003 Beckman Fellowship, 2003 Xerox Faculty Award, 2003 Cottrell Scholars Award, and the list goes on and on. So that's just to give you a feel of what we're talking about when we say we have some of the top researchers in the world here, and we're very fortunate to have Professor Taekjip Ha come and talk to us today about little engines that could. Professor Taekjip Ha.
Thank you very much for your kind introduction. For a big shoe to fill here. Can you hear me in the back?
Yeah.
Okay, good. I have a little cold, so please bear with me. So, in my lab and others doing biophysics in this department and other departments in the country and in the world, we're trying to use physical tools and concepts to study biological systems. And one big motivation is that once you understand the fundamental mechanisms of the biological systems, because many of these systems can cause very serious human diseases, such as cancer if they are defective, perhaps we can find a rational way of fighting the disease. So, perhaps the ultimate motivation is to improve the human condition. More physical motivation, I can list a few things here. Well, biological systems use components that are very small in between 1 and 100 nanometer length scales. I'll explain what a nanometer is in the next slide. And this is a scale of future electronics. So as the chips become smaller and smaller and faster, one reaches a very small limit length scale, and this is a very important length scale for many different things in the modern technology. Second of all, nature has engineered over billions of years to make really excellent, very robust, efficient molecules. Okay? Especially, for instance, bugs such as bacteria. That was my son. Well, he's crying, so hopefully he's okay. Well, so also many of the biological sensors often reach the physical limit of sensitivity, like your ears and eyes. So perhaps you can do ultimate reverse engineering. You learn the design principles of these very efficient, optimized nature's nano machines, and then perhaps use this to make our own devices. So that's another motivation, in addition to biomedical motivation. So 1 nanometer is 1 billionth of a meter. A meter is about 3 feet. Many of the biological molecules are between 1 and 10 nanometers in size. And this is the human hair electron microscope image. Across, this is 50 micron, which is about 50,000 nanometers. We can actually fit 50,000 nanometers in the dimension of a human hair. So we are really talking about very small length scale. Okay? So this is an image. There's images of several cells that are found in the bodies. And they are labeled with different colors to highlight different components in the inside the cell. You can see that it is highly structured. You can see different colors in certain parts of the cell. And to organize a cell in a very specific way, you have to actually move things around. So motion is very important in life. And a lot of these movements are done by proteins called motor proteins that move in the cell and that move in along the highways inside the cell. So I'm going to talk about the motor proteins today. So here's demonstration, not much of a demonstration, but here's a motor protein, and it's moving on a road. And, okay. So one thing to notice here is that unlike highways, this is a one-way road. So a given motor protein knows which way to go, and it only goes in one direction, not the other direction. And we have a lot of cars, Japanese American cars moving on the highway, and they use a fuel, gasoline. That's the energy source. Okay? And you can make the analogy to motor proteins in the cell, and shown here, they're called kinesin, thymine, myosin, helicase, and so on. The kinesin molecules move on a track called myosin. I'll explain a bit more about that later. And myosin 5 is on a track called actin filament, and helicase moves on a track called DNA. And many of you have heard about DNA. And the fuel molecules are, in all cases, small molecules called ATP. You basically burn this fuel and convert it into mechanical movement that moves the motor protein in one direction along the highway. This is a picture of a cell, and you can see two different colors, green and orange. And green is due to actin filament, which is the track for myosin 5 that I will discuss in the first part. And is the orange is a track called microtubule, and that kinesin is walking along. So you can see that these rows are laid down in the cell in many different directions, and they're not completely overlapping. So there's a design also behind how to lay down the rows, so that these motor proteins move around. And this is a helicase that I discussed. It is that moves on the DNA. DNA is made of two single-stranded DNA, and what it does is to move all as a motor and separate the two strands. And helicase literally means an enzyme, protein that destroys a helix, DNA double helix. Yeah, I'll explain a bit more about that later. So we physicists use quite new and state-of-the-art single molecule techniques to study the motor proteins. And one popular inclination is called, and this is actually came earlier than our own tricks we call Fiona and Fred. And in optical trap, what you do is you use an invisible laser focused tightly to a small spot, and then they can grab a small microsphere and move it around. Okay? And then if there's a protein here that is pulling on the DNA, then you can play a tug of war between the protein and then you can measure the movement of the protein on the DNA. So that's the idea. And in the first part, I'll give you an example of using a technique called Fiona to study myosin 5. And the second part, and then I'll take some questions. And then I'll use the second part to describe a technique called Fred, and the use of it to study helicase movement on the DNA. And that's the outline of my talk. So here is artistic movie of a motor protein walking on a track. This is a kinesin walking on a microtubule made by Milligan and Vail. And you can see that the sphere molecule is coming in and out, and the molecule is burning the fuel molecule, breaking it, and then it moves forward along this track. Okay? And there are two forms, two blue structures here, and they are called heads, two heads. And as you can see, the way it's moving, the two heads are alternating. Okay, this one binds here, and then this one binds here, and then later on, this one moves forward to go ahead, and so on. And so it's more like walking, protein walking on this track. Okay, but this mechanism is called hand over hand. It's very confusing because I'll call them feet, okay? But people call them heads, and then they call hand over hand. Okay? So to make it clear what they mean by this, I asked my daughter to enact this movement just upside down on the next slide. Okay, so you can see that she's pretty good in doing the monkey bar. Yeah. And she's quite good because once she binds to the monkey bar, she can do it for several times before she falls off. Okay, that's very important if you want to carry cargoes in the molecular highways, because you don't want to just go on and off the highway. Okay, and she can do that for much longer. In this case, the track is limited. All right, so that's what people thought until a few years ago. Okay, this is how the motor proteins move, just walk, okay, or hand over hand. But Jeff Gibbons in Brandeis University actually pointed out a few years ago that, well, it turns out that all the data are also consistent with a different mechanism called inch over, not inch over inch, but inch-worm mechanism. And in fact, his new data suggested that perhaps inch-worm mechanism is the correct one. Okay? And basically, his model is this. You basically, instead of protein walking, it moves like a baby crawling, so it moves this way. And the reason why he preferred the inch-worm was that, but he thought that a protein is not as good as a human, so you cannot just walk this way without twisting your upper body. He thinks that the protein has to, move this way, okay? Because there's not enough flexibility there. So what he did was he attached like big long rod to the top of the protein, and he watched the movement of the rod. Okay, so if you have hand over hand, you'll see the stringing of the rod, and if you have inch-worm, you will not see the stringing. And he didn't see the stringing, so that was his reason why he proposed the inch-worm. But of course, there are some assumptions. So there was a controversy, and then, in fact, Paul Sevin, sitting over there in the department, my colleague, had an idea to test this, okay, using a very direct method. So I'm going to describe that experiment today. So how did people actually measure the movement of this motor before? Well, they use optical track. So there is a kinesin with two heads, 2 feet, or 2 hands, whatever you want to call them. And then this is carrying a cargo, this microsphere, and it's carried in optical track. And then you can measure this step size very accurately. So let's see if I can click on. So here is an individual laser light in the middle of this crosshair, and then this microsphere containing the kinesin molecule will be moving around and finally trapped by the trap, and then they actually move the trap to land it on a microtubule track, and then they can see it move. Okay, so they show you this movie taken from Stephen Bloch's lab, Stanford University. You get the trap. And then you can see the microchip will track here. And then it moves. Okay? And then once it reaches a certain limit, it snaps back because the force is too strong to overcome and to move away. And by doing this type of measurement, they were able to show that they can measure the position of the beat very accurately as a function of time. And you can see steps, and if you measure the step size, then you can deduce how with what distance the center of rest moves, basically the body moves. Okay? But it turns out you cannot distinguish between the two mechanisms that I mentioned by measuring the center of the body movement. You have to measure the movement of the foot. Okay? So I have another demonstration here. So what I wanted to do was to have a light bulb. Okay. Here's a light bulb, and then you attach it to. Okay, and I have to turn off the room light. Right. And I turn off my screen. Oh, it's very dark. Can you see this? Okay. So if I move hand over hand, then you can see that step quite large. Okay. But if I move instead inch one, then my right foot will move half the step size. Okay. And that will be actually the size of the body movement. So that's what we did. Go back here. So we have to find a way to attach the light bulb to the foot of the molecule. And that's what you do. And then hopefully when the protein walks or crawls, you can measure the step size and then see what you get. So we did that using a very sensitive fluorescence microscope, and I'll explain what that is later. And this protein is known to move by a 37 nanometer step size of the body. Okay? So if the hand is correct, then you'll see the movement of 74 nanometer times 2. For instrument, you'll see 37 nanometer steps per foot. So we use fluorescence, and it's very simple. You shine a laser light as a fluorescent molecule. This is a chemical formula that you should just ignore. Just think of it as a sphere, green sphere, and then it will give you a green photon. Okay? Different color from the laser itself. You can also have different colors for different fluorescent molecules, and you can think of them as the light bulbs. Okay? Different colors, and I also have some solutions contain. Right, okay, so I have some fluorescent dyes in this solution that. So what you do is you just attach one molecule to the foot, okay, and measure. And if you take a video image of such a molecule, you get something this. You get one nice spot, okay, and the size of the spot is given by the physical limitation of imaging, for diffraction unit, and that's about 250 nanometer in size from here. But you can actually use a mathematical function to fit the data and determine the center with a much better precision, accuracy. And it can be done with about 1.5 nanometer, which is really, really good. And that's, again, reaching the physical limit of about 1.2 nanometer. So we came up with an acronym, but this is Princess Fiona in a movie called Shrek. Maybe some of you have seen it. I've watched this movie many times because of my kids. Her name is Fiona, but it turns out her name is an acronym. for fluorescence imaging with 1 nanometer accuracy. And the one is very important because people call a nano technology a nanometer when the object is about 100 nanometer. Okay, but here we are really talking about 1 nanometer. Well, 1.5 nanometer. Okay. So now we can determine the center of this guy as a function of time with 1.5 nanometer. We have done that, and then we plotted the position of the foot as a function of time, and it shows very beautiful steps. Okay, you can see very beautiful steps. If you measure the average step size, we get 74 nanometers, which is twice the step size of the center of the body. And this proves the hand-over-hand, and this proves the initial mechanism. Okay, so this is so far the most direct demonstration of the hand-over-hand mechanism using a very simple but powerful technique. And when this paper was reviewed by a journal, one referee said that, oh, this is really beautiful. The data is so nice, then you should be able to see the movement from the movie itself. Okay? It turns out that the student did not look at the raw data itself. He simply used a computer program to fit the data in this curve, and then he went back to it, and then you could actually see steps with your eyes. Okay? So here's this molecule sitting here, and then you move down the left corner, and you can actually discern steps. So there was a step, another step, another step. In fact, the pixel size is 80 nanometers, so you can see a 700 step as almost a pixel size medium. So this work was done mainly in the group of course 7 sitting over there, and it looks like that. So if you are on the street, please don't do not forget to tell me that you like the work. Okay? All right, so I take a break here and take some questions. Yes.
What molecules or elements around DNA and surrounding? Are there other elements that this is protein type that makes stay involved in the brain? To other molecules that evolve.
The question is, how does the molecule keep moving? There should be also other molecules binding and kicking it around. And this is a great question. And the cell is not a free environment. You have lots of things densely packed. So my demonstration was idealization. In fact, you have to imagine somebody walking within a subway, densely packed. But somehow these molecules know how to move without really getting perturbed. So in fact, what we have done is apply opposing force. Basically, when the protein is moving, then you stab on the neck and then pull backward and see what forces are necessary to stop the movement. You have to apply a sizable force. So they're actually made to overcome sizable opposing forces to carry out their function. So they are extremely robust, and that's one of the reasons why you want to understand them, perhaps use the principles to make our devices in the future. Yes.
So you tell you about the rotational motion indicating that it might be this inchworm.
Yeah.
Now you see that it's hand over hand. How do these things? How do you explain those? Two different, apparently different.
There is a different mechanism that also proposed by Jack Taylor. It's called asymmetric hand over hand. So in that case, basically, as there's a twisting going on, but each swing, you get a little different tension, okay? And while keeping the body constant. There's evidence from other groups that suggest that perhaps hand working mechanism should be asymmetric, but and I have not been convinced yet. Okay? Is that fair? Yes?
So is there kind of a flow of energy to one direction? Which causes the direction?
So why does it move only in one direction? Oh, because it says one way. And the proteins not to be. No, just kidding. Well, in fact, what happens is that the highway has a built-in asymmetry. It's not symmetric, so it has a directionality like an arrow. Okay, it's utilizing that directionality just to be in one. So, there are some proteins we can make a mutation, modify the protein slightly, and then it moves backward. And so it doesn't much to change the movement direction. And in fact, we don't actually, it's not clear whether when the molecule goes backward, whether they actually bind in this way and then still move on, or bind the same way as move backward. It's not known yet. Yes?
What's the purpose of this distinguishing between these different mechanisms? For example, if the method hand by hand is correct, then what confusions should make of the
Yes, the question is, essentially, the question is that what is the big picture? Why should you care about whether it's hand over hand or inchworm? And that is a very good, fundamental, and difficult question to answer. Because I don't have a good answer for that. Because I cannot say, if there's one mechanism, then we can develop drugs against, diseases caused by these proteins more easily. We can't, there's no evidence like that. So it's a fundamental question about understanding how these proteins work. Okay? And, yes?
Is it the hand over hand? Is that a specific method used to unzip the DNA?
DNA unzip is the next part. There is a model is not actually proposed for DNA, mind you.
What do you use to allege that it interferes to the protein?
So the modern biotechnology have worked out wonders. Okay, now we can attach these dyes to the fluorescent molecules more or less wherever you want, in one place very specifically. Okay, and this site neurogenesis. And this came about 20 years ago. It's now so standard, even the lab can do this. Okay, and they do it very, very well. Okay? So it's very standard. We didn't invent the technologies. Okay.
Can I ask one more?
Yes.
These motor proteins, it has its own fuel when it starts this process. In other words, like a battery in that toy. It takes its own fuel with it, or is it constantly supplied with
Is it so? Yeah, that's a very good question. So the motor protein does not have a fuel tank, okay, containing many fuel molecules. In fact, it has only one. Each head has only one binding pocket for one fuel molecule. So basically, the fuel molecule has to be supplied constantly from solution.
So it has a supply train coming in.
Yeah. Okay, so real hard, but because he asked a lot of good questions, I'll give him a Physics Society t-shirt. Okay, so we have this Fiona to study this myosin 5. The next question is that, can you do similar things that move on DNA? That's my main topic. So, how many of you have seen DNA as a double helix? I mean, can you imagine if you know DNA as a double helix? So this is picture taken from this very famous paper by the Nobel laureate for this discovery, who discovered the DNA structure, published more than 50 years ago. And in this paper, they wrote that it has not escaped our notice that the specific pairing between the two strands of DNA, base pairing, suggests a possible copying mechanism for the genetic material, because one can act as a template for copying to make more DNA. And this statement actually is considered by many to be the understatement of the century. Okay? Because it's a really powerful statement, but they really disguise it in a way that, in a very shy way. In fact, these two guys got really worried after they published this paper because they didn't make it very explicit. They worried that somebody may actually write it down in a very explicit way and claim the credit for the discovery. Okay? So months later, they published another paper here, okay? In April, May, and to propose this specific mechanism of base pairing to copy DNA. But at the end of this paper, well, it is much to do with this, and in particular, what makes the pairings unwind and separate? Because this double helix is a very structure, okay? And but if you use the DNA, you have the same. But how does it achieve this? Okay? And 6 people first hit basically, and they indicate the enzyme that the function to unwind and separate 2 strands. Then there are thousands of helicases have been discovered, and that's what I want to talk about here briefly. So the most fundamental function would be unwinding the DNA. So let's say this is the DNA double helix, and you need to unwind the DNA before copying the DNA replication. So helicase goes first and then unwinds the DNA, and then copy the DNA into 2 daughter DNA molecules from mother molecule. And the helicase unwinds the DNA and also moves along the DNA, so you can call it a motor protein. And its fuel molecule is also ATP. It needs to burn it. In our genome, human genome contains more than 100 helicases. So, clearly they are not just doing this, they are also doing many different things. Some are very highly specialized. Some can cause very serious diseases, including cancer, if they are defective. So there are many reasons to study their mechanisms. So one question is, if you think of it as a motor protein, can we also measure the step-size helicase movement, just like you did for myosin 5? So my answer to that for now is that we haven't done that yet. Okay, so we are going toward that, but we found something interesting along the way. So just to compare the step size, myosin 5 takes a 37 nanometer step per ATP burning, and kinesin is another protein that moves by 8 nanometer. And DNA RNA polymerases, they are enzymes that copy DNA and they make RNA, and they move by 1 base pair step that is 0.34 nanometer. So it's very small. So even with Fiona, with 1.5 nanometer accuracy, we cannot measure. Okay, it's 5 times smaller than that. Okay, and helicases we don't know yet, but it's somewhere between 1 and 10 base pairs per ATP. And clearly, this is still very challenging to measure using Fiona. So we need a better technique to measure even smaller movements. And that's the technique that we use. We use single molecule FRET. The FRET is an acronym for fluorescence resonance energy transfer. So what it is that you have 2 light bulbs instead of just one light bulb. You have 2 different colors here, okay? And initially what you do is you just, you want to help? Yeah, so she's going to help me here. So you excite the green light bulb using your laser. And when they're far away from each other, then you only see the green light, okay? But when you bring one close to the next, then what happens is that green gives energy to the red, and then you get the red signal. Okay, when you move it farther away, then you get the green back. Okay, and you can do it back and forth. I can do it very sorry. So you get the idea, okay? But imagine looking at this not this close, but from a mile away. Okay, then you cannot see these two as two light bulbs. You just see it as one light, but different color. Okay, that's what we do in our experiment. Okay, what we do is we attach. I think, yeah, great, thank you. Yeah, we attach these light bulbs to different parts of the molecule, and you measure the color from far away, and then based on the color, we can deduce the distance, and we can deduce the dynamic changes. And here, this movie illustrates one illustration of a dancing DNA molecule that is tethered to a surface. And the dyes that we use are called Sci3 and Sci5. Again, the detail is not really important, but I just want to mention that, in fact, our human genome was sequenced using these dyes, okay? And they're also very good dyes for doing single molecule measurements. So, you know, what is good for one thing can be good for other things. So the way we just illuminate a large area of the sample on which you attach many your molecules, and then use a camera to detect green and red colors simultaneously. In fact, you can see single molecules here and here of different colors, but in fact they correspond to the same set of molecules as you can see within the circle. And so what you do, attach our molecule, which is a trap on the surface, and make sure that the surface is not sticky toward the proteins, and then we add the protein, and then let the protein do the job. There's a movie that shows is an RNA that goes between two shapes. In one shape, the dyes are very close to each other, the red signal mostly, and the other shape that are far from each other, you get mostly the green signal. Okay? And now you have more than hundred molecules on the surface. Each dye is from a single RNA molecule. And you can see some of the frequency, green and red, because the molecule is going back and forth between the two shapes. So you can do this shape change measurement over quite a long time from many molecules at the same time. And what we can also do is to just go in to one molecule and then take the intensities of the green and red light as a function of time, because we have a movie here. And we can plot that as a function of time shown here. So green and red. Let me apologize first if some of you are colorblind, because I have green and red. In fact, two of my best students are colorblind. The only problem is I have to make my own slides. They cannot make my slides for me. So you can see that when the molecule goes back and forth, it's going up and down in an accurate manner. The green goes up, red goes down at the same time. You can also calculate the Fred coefficient, which is high when red is high, low when green is high. So basically, high Fred value means that two dyes are close to each other, and low means two dyes are far. So that's all you need to know to understand data I'll show you next. Right, so let's try this if it works. So we're shining the laser light, 3 tubes. And the first tube has no, just water, so you don't see any fluorescence. Second tube has this molecule, DNA molecule that has two dyes very next to each other, so you get the red signal. So you can see the red color here. Okay. And the third tube has just the green molecule, so it's only green. So what I'm going to add the helicase together into that middle one, and then and demonstrate that helicase can unwind the DNA using ATP. The color will turn green. Okay. Well, I should put ATP here, but. Steady hands. Right. Can you still see it? The map? Yeah. She's going all over the map. Yeah. I'm taking. So this tube contains a helicase plus ATP. I'm going to add some amount into the middle one. And then use this to shake it. Did the color change somewhat? I think it's changed. Okay. Here I demonstrate fret, okay, and helicase function, and using ATP. Okay. So that's the demonstration. Thank you very much. The helicase that we study is from a little bug called E. coli, from helicase, but this is not really important. But these findings, based on our work and some work, show that one motor protein, okay, can move on the DNA, on the single-stranded DNA, but cannot unwind DNA. Okay? You need to have at least two, okay, to combine, and then they can unwind DNA together. So here's a little animation. There's a motor protein moves to the DNA, and each duplex DNA, it cannot unwind the DNA as alone. And then binds and then moves toward it, and then magic happens. They transform into protein, as I call it, a monster truck, and then you can unwind DNA as a tool. So I call it monster truck mechanism. So this was known before. So one thing to notice that one protein cannot unwind DNA, while it can move on the DNA. There's a motor protein, but cannot unwind DNA. So what's the good use for it? Okay, if you cannot unwind DNA, and it turns out that we have a very small number of these proteins in the cell, that it is unlikely that this monster truck will actually form in many cases, okay, because you need to have two of them coming together. So there may be a function of this protein that derives just from one molecule moving on the DNA. That's what I'm going to suggest in the next few slides. So what we wanted to do is the following. We wanted to put the green molecule on the protein and red on the DNA, and use FET to measure the relative movement between the two. Okay? And this can give you actually much better resolution than even Fiona, because this is a relative distance changes. So this is what the data will show. There's a protein in solution, and we are exciting the DNA, this area, using the laser. So this is not visible initially. And the protein is moving around, and in solution but it's not visible. So the signal here is very low for both for green and red, because you don't excite the red directly using a laser. And then it protein is around, diffusing around, and finally binds the DNA, lands on the track. And then you can use ATP to move along in this direction. This is one way again, like gradually. And that's what we see, especially as a function of time. You can see the protein binds, the signal suddenly shows up, green and red goes up, and then gradually green goes down and red goes up, and you can see the efficiency calculated from this data is increasing gradually. Okay, so what you see is a binding and moving toward the red line. Exactly what you expected out of experiments on this protein. But it's good to see that from this kind of experiments. So that was already a bit exciting for us because you can actually use a single molecule flat technology to measure this movement. But what was actually more exciting and more strange or more protruding or unsettling was the fact that it doesn't end there. I mean, it doesn't end there; it actually continues. And as I show you, what happens is that so as it goes to high fret, it suddenly comes down to low fret in one step, and then gradually goes up again, comes down, goes up again, comes down. So you see this gradual increase in fret, sudden decrease, and gradual increase in fret, sudden decrease. Okay? So that's very strange and like that. You see in fact the same measurements in different lengths of DNA here. If you make the DNA shorter from 80 to 60, frequency of this behavior increases, meaning that this increase is due to really the movement of the DNA. So we call it sawtooth. Okay, so it looks like that. It's beautiful as this, but just to illustrate the point, actually what we discovered here was that this moves, but suddenly it's back, then moves, snaps back, and moves down. Yeah. It's very because if you think about it, this car is moving along this way, and suddenly a battery came out. Here and then it goes. So it's really strange. Okay, and how that happens is also that I may be able to tell you a little bit if you. So that was really exciting. This has not been reported in the scientific community yet. So I call this molecular Sisyphus. And do you know who Sisyphus is? Yeah. Yeah. So it's a Celtic Greek myth, and here the god, the supreme god, Zeus. So basically every morning he has to get up and roll this rock up the hill, only to see the rock fall down again. Okay, and then next morning he has to do it again and again. So it looks just like that, just like our data.
Should it be waiting for his other partners to get there, so there's two of them?
Here's a question. Okay, I can maybe address that later. All right, so I think this is a very cute metaphor, but this Sisyphus metaphor is resoundingly unsatisfying because we think we found something very novel and interesting. Yes, question?
Can you demonstrate how the protein moves again? The sawtooth, how does it connect?
This one?
Yeah. Does it jump?
Okay, so this is our data tells us because within our time resolution of 20 milliseconds, it is instantaneous. Okay? Exactly how it does that, we're not sure yet. But I can speculate later on. So anyway, so it's cute, interesting metaphor, but unsatisfying because we're excited that we found something very novel and interesting, but if he's doing this for nothing, okay, then maybe it's not that interesting. So is there any meaning in the suffering? And in fact, a few weeks ago I gave a talk in MIT while Boston Red Sox was fighting against New York Yankees. And so I wanted to actually come up with a joke about Red Sox in the talk, but I couldn't come up with anything. But I know what I should have done. It's this. So this is, it says in Boston, okay, Sisyphus again begins pushing the rock up the hill, intent on finally getting over the top while hoping to not get crushed when it begins to roll back down. So this was spring this year, just before the Major League Baseball season started. MLB.com. Both Boston Red Sox not winning the championship for the last 86 years, mainly because of New York Yankees. You know, every season, the team begins to work, and then later on, the New York Yankees just push them back, and so they have to begin the next season and so on. So is there any meaning to the suffering? So this is an ad from Nike. So it starts from 1990 when they didn't win the World Series last time. It moves on. 1950. They're still waiting. My point is that to me, these fans, they don't seem to be suffering. They're actually enjoying it, and there's a meaning to it. I mean, maybe it's very therapeutic, So in fact, after they finally won the World Series, in New York Times there was an article by this guy, and it ends by saying this: the truth of the matter is that through all those 86 years, we weren't really miserable at all, because although you're not winning the World Series, you're not unwinding the DNA, okay? But this process itself was meaningful, okay? So my question is that in our experiment, can we find a meaning about this process itself, okay? So here is our biological spin, if you want. This is the template DNA, your mother DNA, that is being unwound by the helicase, but not our helicase. And then the red DNA molecules are newly synthesized DNA molecules based on the blue template. Okay. It turns out that very often this replication stops because of damage on DNA. Then you have to take apart the proteins and then repair the damage and restart. You have to restart patient after it's stored with the DNA damage. And it's known that our helicase is involved in this process, the replication restart. Okay? It is also known that our helicase binds to this structure very tightly. So our thought was that maybe what it does is to bind to such a structure, and then this single-strand DNA is a one-way signal for the helicase to move in this direction. Okay? So it moves along this direction, but it hits this barrier. Okay? Because the double strand, it cannot unwind the DNA, and then it somehow hops back, and then goes on again. Okay? Goes back and forth, on and on. Okay? So, and it's also known that these kind of proteins can keep other proteins away from it. So our thought is that perhaps the mechanism is not in molecular Sisyphus, but molecular dental floss. So, but in the reverse way. So what I'm talking about here is the following. Well, it's not the dental floss, the single-strand DNA that is keeping the teeth of the helicase clean. It's the teeth, okay, that is keeping the dental floss or DNA clean junk or the other proteins until proper molecules come and bind and restart replication. So that's our spin to it. In fact, I made exactly a structure that looks like that, as shown here, and we have done the same experiment, and we saw the same kind of sawtooth in this case, going down gradually, coming up suddenly, going down gradually, because it spines, moves away from the red dye gradually, and then suddenly snaps back, and so on. So maybe it's not just the image, maybe there's something real about this. So I want to summarize the main parts of the talk. So we have discussed two techniques for Fiona and Fred using fluorescence, single molecules. Myosin 5 moving water protein moving on filament and shows that it moves hand to hand. And using Fred, actually show some interesting data that we call molecular Sisyphus or molecular flaws, whatever you want, which aspect you want to emphasize. So this is just an illustration, how these novel new tools can be used to, address existing questions, which is in this case. And in this case, you just find something, entirely unexpected. So then the new questions that arise that you want to now follow up. But I want to show a few slides about it. What to do is now building fluorescence with optical trap so that we can pause and then measure the So this is the RNA molecule that we studied before using these two dots. And usually we are just looking at them passively, as they just fluctuate in open and closed shapes. But what you want to do is now to add a DNA handle and a microsphere and an optical trap, so we can grab on the sphere and then pull on it. Okay? Actually, you can stop in this direction and see and still measure the DNA movement by fluorescence. Measure how the molecule responds to external force. And this is another question relevant to your question. Basically, in the cell there will be experiencing a lot of different kind of forces, and how do they actually function? And you'll be able to answer these questions using this type of experiment. That's the wrong slide in the wrong place. Okay, the wrong time. Okay. The second one is we want to now move our observation, glass surfaces into living cells. Okay. Because that's the native environment for these molecules. But it turns out that what they use, they are not really good enough to do kind of measurement that we do, that I showed you, because they are not bright enough. They actually die quickly. And it's called quantum dots of different colors, and they're very bright. They last forever. The problem was that they were known to blink. Okay, and it's example is shown here. If you measure the intensity of a single quantum dot, it's 20 times brighter than a single diamond molecule, but as a function of time, it goes up and down. So it turns on and off. And you don't want to take it using a flickering light source. It's like making a movie on the stroboscopic illumination condition. And it turns out in our lab, we found a condition to completely to remove the blinking, so you get very stable emission from such a quantum dot. Then now we can really imagine use that to do the kind of measurement that we do now, but in living cells. And this is just a movie of single quantum dots diffusing on a biological membrane. You can see they are moving around. The movie lasts forever. They don't. They can go on and on. So which is really neat. The last topic that I want to mention is the fusion with nanotechnology. It's still a very long, far away goal, but if you want to eventually use the biological molecules to build devices, perhaps one can start thinking about these issues. So here is a kinesin molecule moving on the micro. But you can also reverse this. You can attach kinesin molecules to a glass surface, and then many of them can cooperate, slide microtubule on the surface. So it's a skin. So the kinesin molecule is now stuck on the glass surface. It's a microtubule. And this labeled with a red colored dyes. So you see that as red actually lines. And then they can collectively basically move, slide the microtubule on the surface. And this is the work done by a group in Germany. What they have done is they also attached a long molecule to the microtubule, and they stain the DNA molecule using green dyes. Okay? And the next slide, the next movie that you see this microtubule in red moving on the surface powered by kinesin molecules, but carrying the DNA cargo in green. see. So in this movie, the red one is the microtubule, okay? And green dot, they're usually coiled up. Because flexibly they're coiled up, they appear as balls. And then so this microtubule, red one, is 6 DNA molecules. It's kind of cool. This next movie. This red one is carrying the DNA. The middle is the red moving. It pulls the DNA and stretches. And this is even funny, more funny, even funnier. In this case, 2 microtubules grab on it, and so the DNA is getting pulled. Until it breaks. And this one is also interesting. So you can see that people are just playing. So this DNA is attached at one point to a surface. So actually, now this microtubule is making an arc. Okay? And then it breaks off. The final movie here is making actually even smaller arc, where it's sort of rotating on and on. So, if you ask me what they're good for, I don't know. But you can combine these nature's molecules and molecules in a way nature never does, and maybe combine new things. All right, so to conclude by thanking the group of the work. So Suamiam, Ivan Rasi, and Sang-Cho Hong, and Sean McKinney are the people in my lab, and as Ahmed Iris is a graduate student who did the myosin 5 experiment. And also that experiment was a collaboration with Yale Goldman's lab in Pennsylvania. And the helicase work that I showed you was a collaboration with Yale Goldman's lab in Washington University. And just the one last thing that I want to show you is that what scientists do after hours. Have you played this game called Yeah. So I heard that it's a game developed by Russians in the Cold War period, so that Western scientists play this game so that they cannot do science anymore. Traps. So they have to meet at the same time, and then you can control them using computer software and a game of Tetris. The music is from independent version of this game. Anyway, so thank you very much. I have another t-shirt. So.
For questions, if you are leaving, please be quiet so the folks who are asking questions can have a chance to ask some more questions.
So the question was, what is movement? The fundamental steps for the myosin 5 and helicase. In the movie that I showed for myosin, slow down the movement by reducing speed. So that's why it's slow. If you go at maximum speed, it will take about 10 milliseconds. 10 milliseconds. So the 100 steps. So the question is that when you see the molecule hopping back, you can see that when it actually due to another molecule and doing it. We thought about that possibility, and we have done many experiments to rule that out. And I think one of the most important experiments that we did was when this was going on, we actually turned on the flow system to wash away all the free proteins in solution. Okay, but it still goes on. Okay. Yes.
Let me stop your problem. Is there possibly any kind of electrical charge that resists for reach? Is there any kind of electrical?
That is a very, very good question, and electrical, basically, charge attraction repulsion can be a very important effect. Not sure if that is relevant for the surface itself, but DNA is actually charged negatively. The protein that we study, helicase, is charged positively. So there is a tendency to bring them close to each other. Okay? And so what I think is actually happening in terms of stepping back, we're not really sure about that yet, but so if this is the DNA that is, that we are moving along, so I'm moving on the helicase, I'm moving on the DNA, and I hit again, and then there's somehow a signal that tells us helicase to reach back. Oh, okay. And then it jump back, and then do it again. Okay? And there's some data that suggests that. And of course, in that case, maybe in the helicase there is a positive surface that is responsible for preventing that. That is possible. So the question is, how do we immobilize the DNA on the surface? And my answer is carefully. I can more details at home.
You say again when you see that snap back, what that time is, or what your resolution is, if you're instantaneous. So.
Yeah, so the resolution for the experiment is 20 milliseconds. So that's going to be 1/50th of a second. And that is very similar to the resolution of the time scale of the video. Because in video you have 30 frames per second, 33 milliseconds. So that's about our time resolution. And within that time resolution, it appears as an. Yeah.
Have you done the scientific experiment with more than one type of helicase?
More than one type of helicase?
Yeah, helicase.
No, we haven't done that. Because one reason is that we have to explore a year of protein engineering to make a protein with only one dye. Okay, and we are planning to do that for different helicases. And it'll be interesting to see whether this is functional, many different helicases, or function specific to this helicase. We are not sure. That is a very good question. Yes.
Do you ever work closely with experts on PCR to maybe answer some of the questions, problems that come up with polymerase chain reaction, plant science, or microbiology?
Yeah, PCR is an acronym for polymerase chain reaction, and in fact, it was discovered, invented in the early 80s, and resulted in a Nobel Prize because it was so popular. Without PCR, there's no human genome sequencing, there's no attaching dyes to proteins. Everything was possible because of PCR. So actually, we are using the technology all the time in my lab just to make the molecules to study, but we are not really using that itself, using PCR itself to research. Yes?
Dr. Lugo, main topic, but could you briefly explain how the chemical reaction actually drives mechanical motion?
The question is, how do chemical reactions drive the mechanical movement? That is a very good question and fundamental. Okay. And in fact, I think many people trying to understand the coupling between mechanical work. Okay? And what's really amazing about some of these motor proteins is that myosin molecules are typically efficient in about 40%, meaning if you 100 fuel units, then you can actually convert 40% into mechanical work, which is much better than what the human-made engine can do. But also there are proteins that rotate. Okay? People claim that these proteins function with more or less 100% efficiency in converting chemical energy into mechanical work. Okay? And so the question is, are there a general mechanism for every protein, every motor protein, or is it specific to each motor protein? I mean, this is a question to be answered in the future. But what this makes, the high efficiency of converting makes, decided if you actually learn how to do it, then maybe we can also make our machines that are as efficient.
What do you think? Tough audience. I have some good.
I'm not sure. I'll let you decide who should win the T-shirt. I like it. So, what?
Yeah. The last question, right?
Last question, yes.
If you have more questions, I'm sure Taekjip would be happy to talk to you down front. Let's thank you once again.